Now if you look into this diagram, because I have already explained to you, so this is a male and female. Okay, so the male and female, let me change the color code here. So the female the females is actually in a mitosis, meiosis 1 and meiosis 2, we know that. So all the female eggs is in the meiosis 2 arrest stage. As soon as the sperms comes, they gets activated. After this, there is a DNA replication take place. At this stage of the DNA replication, your first mitotic division is taking place. After this mitotic divisions, then you are getting a 2 equal size cells. Okay, so 2 equal size of the cells is made. After these 2 cells, you get a 4 cells, 8 cells, 16 cells and 32 cells, and then you are basically develops a blastocyst, which is roughly around 100 cells or so. Now there are several processes which is occurring, as you can see here. First is the cleavage, which involves, as I mentioned, 2 equal size. Then at the 8 cell stage, that is the process of the morula, that which consists of like 16 to 30 cell stages, the cells become polyhedral. They start compaction is taking place. Okay, at this particular stage, then the cleavage which is taking place inside the cell. Now the cell volume, the volume is less, that's why the compaction is taking place. After this, 2 layers of the cells is making, as you can see here, if you focus here, the 2 layers of the cells which is making a tight junctions between them, that is the outer and the inner layer, and then you get a blastocyst cavity. So this is little bit about your embryonic stages. Now if you look into the outer layer of these cells, which is still not implanted, is all these processes is occurring in the fallopian tube. Now after this stage, if you see, there is a trophoblast, and we see now you understand that why that definition of trophoblast is important. They are making this tight and gap junctions. After this, they lose the gonoplacida, and then they get implanted into the uterine line or the uterus. Okay, so that's what the blastocyst then hatches out into the gonoplacida, and then they enter into the mother wombs, and then they are attached. And just before the process of the implantation, the cells are differentiated into the 2 layers, and that's what is your epiblast and your hypoblast. So what is epiblast? As again to recall, primary ectoderm. Later on they becomes the whole organisms is developed from this, and then the hypoblast, which is basically your primary endoderm, which has been then. After this, the 2-layer embryoblast is called as a bilaminar germal disc. Now if you look into this process, that after this process, if you see, I will come back to the slide. This is, you can see here, let me change the color code. See, this is your bilaminar embryonic disc. So this is why bilaminar, because you have a very distinctive epiblast and the hypoblast. The epiblast, which later on makes the whole organisms, the hypoblast, which later on helps in forming the body excess types. Besides this, you also see here, this is the amnion, and you also see this is the amniotic cavity where the embryo starts developing, and this is like the uterine wall, and this is the blastocyst cavity. This is at the time of the implantation. Now before the implantation process, like coming to here, this is the before implantation process. This is the functions of the cells which is doing. Now if you are driving certain cell types, which is called as the inner cell mass, and that's what term giving inner, because it is inside. This inner cell mass also consists of certain specific cell types which are called as the embryonic stem cells, and these embryonic stem cells will be equivalent to your epiblast only. Okay, that's the epiblast from where you are driving your stem cells. Okay, if you look into the differences between an epiblast and hypoblast, because this term is often coming, they have 2 layers of embryonic discs. Okay, 3 germ lengths, that is ectoderm, endoderm and mesoderm. This is the second layer of the embryonic disc, which we showed in the previous the next picture which you saw. It forms the yolk sac, okay, and it also forms the what is called as the axis, the polarity of the embryo which has been determined. Primitive, it is ectoderm, it is primitive endoderm, which are outer layer. This is the inner layer. This is the embryo proper, and this is the yolk sac driving its nutrients, and this is the columnar cells. There are cuboidal cells. They lies above the hypoblast and they lies below the epiblast. Like for example, if you see in this particular here, this is your hypoblast and this is your epiblast, and that's how you are driving. They are all go at rapid multiplication. If you look into a this is the from the mouse model, and this mouse at the embryo day roughly around 7.5 days, you will find out that there are amnions. Okay, the mesoderms, the head process starts coming here. So this is in the uterus which we are talking about. Now if you zoom into this particular region, you will find that this is your allantois. You also find that there is a very distinct niche of the cells which is called as the primordial germ cells. These primordial germ cells is responsible for the gamete formation, but they basically make a very distinct in the environment. And in fact, based on these primordial germ cells, several of the scientists and the research scholars, they believe that the adult stem cells, because still we do not know from where the adult stem cells comes, but they're saying that during the process of the differentiation, they also acquire a particular niche, and in this niche area, that's the stem cells is been present. Now I think you know that this allantois is basically is making the umbilical cord blood vessels, and it is also making you can say the posterior wall of the yolk sac. So this is a very clear-cut definitions which you are seeing. Now the process of the gastrulation which is taking place. Now if you see, this is making the ectoderm, endoderm, which is all coming from your epiblast, that is your proper embryo. The others are all the supporting cells or providing the protective layer or the nutrients which is there. So now from this ectoderm, you are getting your nervous tissues from your and the skin tissue. That's why your skin stem cells or nerve stem cells which is coming. The mesoderm, this is the probable hypothesis for the adult stem cells. Bone marrow or the hematopoietic stem cells or muscles and bones, which gives you tissue stem cells. Then from your endoderms, the visceral organs which is been made, you are getting organ specific stem cells, or esophagus, stomachs and intestine, which is making, that is the intestinal stem cells is derived. Now at this time, you are also getting a primordial germ cells which is very distinct streaks or different niche, and from this you are getting your gonads and as well as your germinal cell which produces the gametes. Now in the same this is a part 1, and the part 2 is what has been shown, that is the tissues which has been derived, and you also get certain multipotent cells, and this multipotent stem cells is where you are getting your neural stem cells, your hematopoietic stem cells and organ stem cells. Now what they do, probably we will see in our next video. I know this is a comparatively little bit longer video, but to make you understand the concept that from where the stem cells is coming, I hope it will be beneficial for you, and you can retain with the definitions of the cells, what are the cell types, from where you are getting the embryonic cells, from what is the epiblast, and how we are getting the adult stem cells. I hope this makes a little bit difference to you. If it does, then please subscribe to the channels and share with your friends, those who are preparing for CSIR NET examination. Okay, if you have any questions, then you can put in the comments box as well, so that I can come up with a query for that as well. With this, I will stop. Thank you very much.